Welcome back, friends. In this video we shall go to look on the dark reactions of photosynthesis. Normally these are not called dark reactions, but they normally call them as light independent stage. So in short, I can say that they don't call them dark reaction because they don't take place in dark. However, they need the products from the light reaction so as to proceed. So sometimes some of the teachers, they can ask you questions to confuse you. Some of the teachers, they say that they are called light independent reactions, and it's not true that they are light independent. Why is that? Because they depend on light but not direct, it is indirect. The light dependent reaction, they depend on light direct, but light independent reactions, they depend indirect on light. So what you can say that the light independent or dark reactions, they take place in the stroma of the corpus. That's the site for the reaction that is the site for the dark reactions, and they do not require light and use energy, that is ATP, and reducing power from reduced NADP produced by the light dependent reaction to reduce carbon dioxide. And when the carbon dioxide is reduced, they produce carbohydrate. So what you can see is that the light independent reactions, they do not depend on light direct, but they depend on light indirectly. So the products produced in the light reaction, they are used in the dark reaction for the production of carbohydrate molecule. So the use of the term dark does not mean that the reaction is necessarily take place in dark or during night, but it simply means the reaction do not require light direct, may proceed even without light. Why may proceed even without light? If the products from the light reaction such as ATP and reduced NADP, this is reduced NADP. If the product is such as ATP and reduced NADP, they are present, the dark reaction can continue even without light. So it does not depend on light directly. That's why it is simply called as dark, but it doesn't mean that it takes place in dark. However, it uses the product of the light reaction. So the reactions are controlled by enzymes, and the sequence was determined by Calvin-Benson, hence called the Calvin cycle. So since Calvin-Benson was the first scientist to investigate on the reactions of the dark stage of photosynthesis, they are called as Calvin cycle. So if you go to read the informations in a biological science book, you will see experiments which was done by the Calvin-Benson until he came with the Calvin cycle. But that's out of our scope, so we will not cover the parts of the experiment he did, but we will cover the reactions that he investigated, or the reactions that he knew after his experiment. So what is the Calvin cycle? The Calvin cycle is the mechanism of the dark reaction in C3 plants. Later we shall see what is C3 and C4 plants. So in these plants, carbon enter the Calvin cycle in the form of carbon dioxide and leave in the form of sugar. The cycle spends ATP as energy source and consumes NADPH2, that is reduced NADP, as reducing power for adding high energy electron to make the sugar. So as I told you that the dark reaction requires ATP and reduced NADP from the light reaction. So these, they are used in the Calvin cycle. In the dark reaction, complex molecules of starch and sugar composed of a carbon, hydrogen and oxygen are being made from simple molecules of carbon dioxide and hydrogen ion. The sequence of events is as explained below. So below here, there are stages of the Calvin cycle, or sometimes they can ask you the stages of the dark reaction, or mechanism of the dark reaction, or mechanism of Calvin cycle. So all of these questions, they have the single answer. If you're asked about mechanism of dark reaction, mechanism of Calvin cycle, stages of Calvin cycle, or stages of dark reaction of photosynthesis, if they don't specialize to you that it is the dark reaction in C4 or C3, just answer the Calvin cycle. So Calvin cycle proceed in major 3 steps. The first step is acceptance of carbon dioxide, or sometimes it's called as carbon dioxide fixation. So carbon dioxide is in the atmosphere. The plant require to fix it, require not only to absorb, because the carbon dioxide penetrate even in the sponge mesophyll of the leaf, but the plant require to fix it, means it requires some of the kind of molecule. That molecule can bind with carbon dioxide, and when that molecule bind with carbon dioxide, it allows the carbon dioxide to enter the Calvin cycle. So what we are saying here is that the carbon dioxide acceptance molecule is the 5-carbon sugar, that is pentose. 5-carbon sugar, they are called pentose, as how we discussed in biochemistry. So that pentose is called Lipids biphosphate. Lipids biphosphate, or in the short form we can call it as RUBIP. So the enzyme involved is RUBIP biphosphate carboxylase. RUBIP biphosphate carboxylase. It is shortly called as RUBIP carboxylase, or sometimes as Rubisco. And the process of addition of carbon dioxide to a compound is called carboxylation. So the enzyme catalyzing the process of carboxylation, they must have the name carboxylase, as we shall discuss later, as we discussed in the topic of biochemistry. So the 6-carbon product formed is unstable. Here, if the 5-carbon sugar, that is pentose Lipids biphosphate, fix 1 carbon dioxide, and that 1 carbon dioxide molecule, it has only 1 carbon, so it will form a 6-carbon compound. That 6-carbon compound formed is unstable and is immediately broken down into 2 molecules of 3-carbon acid, and these are called glycerate phosphate. So the 6-carbon product formed is unstable and it is immediately broken down into 2 molecules of 3-carbon acid, that is glycerate phosphate. This is the first product of photosynthesis. Remember, this is the first product of photosynthesis. It's called glycerate phosphate, and this is an acid. As you can see, it is 3-carbon acid. The first stage occur as per the reaction below. So this reaction summarizes our first stage. Here we have RUBIP, and then it absorbs carbon dioxide here, and then we have water. Then here we have RUBIP carboxylase. In this arrow here we have an enzyme RUBIP carboxylase. I forget just to put it here, but I will add this. We have RUBIP carboxylase here, and then the product formed is 2-GP. Here, as we told you, that there is a 6-carbon compound formed here in between, but it is soon broken down into two molecules of 3-carbon acids, which are glycerate phosphate, or sometimes they can be called as phosphoglycerides. So this is 5-carbon sugar, and this is 3-carbon acid. This is Lipids biphosphate, is 5-carbon sugar, and this is 3-carbon acid. So sometimes you can say, why are we using sugar again to make acid? This is because we are using sugar at first stage, but later in the last stage this sugar will be recycled again. So that single carbon which we absorbed here as carbon dioxide is the one which is used to produce carbohydrate, but this RUBIP will be recycled, will be regenerated later in the other stages of the Calvin cycle. So it's something to note here that glycerate phosphate is sometimes called phosphoglyceric acid, PGA, or sometimes called phosphoglycerate. That's the same molecule, and it's the first product of photosynthesis. Remember, it is the first product of photosynthesis in C3 plants, but not the first carbohydrate, because glycerate phosphate is not a carbohydrate, it is an acid. So the second stage of the Calvin cycle is the reduction phase. As we know that what is made in the first phase, glycerate phosphate, is an acid. So in the reduction phase, we are taking hydrogen and the energy from ATP. We are using that energy to reduce an acid into an aldehyde, because we know all monosaccharides, they are aldehyde or ketones. This is our knowledge from biochemistry. So glycerate phosphate is a 3-carbon acid. It contains the acidic carboxylic group. that is COOH. And the reducing power of reduced NADP and energy of ATP, they are used to remove oxygen from glycate phosphate, and the process of removing oxygen is called reduction to form triose phosphate, that is, or it is sometimes called as glyceraldehyde phosphate. So triose phosphate is sometimes called as glyceraldehyde phosphate or phosphoglyceraldehyde, that is PGAL, because this phosphoglyceric acid, it is PGA, but triose phosphate, which is phosphoglyceraldehyde, it is PGAL. The L is already there, meaning that it is an aldehyde. So, which is a 3-carbon sugar containing an aldehyde group, which is C single or then H. So, what is done here in the second stage is the reduction from the carboxylic group into an aldehyde group using the reducing power of NADP and some of the energy from ATP. And remember, all of these, they are producing the light reaction. That's why we are saying that the dark reaction cannot proceed in the absence of the light reaction. So the reaction takes place in 2 stages. In the first stage, using some of the ATP producing the light dependent reaction, and in the second, using all the reduced NADP produced in these reactions. The overall effect is to reduce a carboxylic group to an aldehyde group. The product is the 3-carbon sugar phosphate, a triose phosphate. This contains more chemical energy than glycate phosphate and the first carbohydrate made in photosynthesis. So the first carbohydrate, remember, glycate phosphate was the first product of photosynthesis in these 3 plants. But the first carbohydrate is triose phosphate. So the reaction in the first stage, we are using some of ATP. That means there are ATP which are remaining in this stage and they are used in other stages of the carbon cycle. But in the case of reduced NADP, here we are using all of the reduced NADP. So if you ask about the uses of ATP in the reactions of photosynthesis, the first use of ATP is this, the reduction phase of the carbon cycle. It is used to reduce glycate phosphate into triose phosphate. And if you ask about the uses of reduced NADP, it is the reducing power is used to reduce the glycate phosphate into triose phosphate. But this is our reaction. But in the last stage of the carbon cycle is the regeneration of carbon dioxide acceptor, that is Ruby. As I told you before, that the carbon dioxide acceptor is the 5-carbon sugar molecule. But what we are making later is also a sugar. For example, here the 3-carbon acid is converted into sugar by reduction. So what we are making, the last reaction is also, the end product is also sugar. But what we are saying here is that some of the triose phosphate has been used to regenerate the ribose phosphate consumed in the first reaction. The process involves a complex of reaction. It is here that the remaining ATP is used. This stage can be summarized by the reaction below. So the reaction below just summarizes. As you can see here, the things are not very much clear to show this reaction, but I try to adjust so as you can see well how that this reaction takes place. So the reaction looks like this. Here is an ATP. Okay, so the reaction looks like this. Here we have ATP, and then here we have ADP. The phosphate group is used to convert the ribose monophosphate into ribose bisphosphate. Monophosphate means it has only one phosphate, and here it have 2 phosphate. So As we are going to finish this lecture, we can see about the fate of phosphoglyceraldehyde, PGAL, or triose phosphate. What is the fate of the products of photosynthesis? Where are they going? So the first thing is that they are using synthesis of other carbohydrates. Sometimes this question can be asked, what are the uses of the end products of the photosynthesis? So the first thing is that they are used in the synthesis of other carbohydrates. For example, taking triose phosphate, it is the 3-carbon sugar. You may make glucose, that is hexose, because triose phosphate is the 3-carbon sugar. You may make glucose, then from glucose you may make sucrose, that is disaccharide. You may polymerize to form starch, you may polymerize to form cellulose, and etc. etc. Another thing is the synthesis of lipids. Lipids are mostly esters of fatty acids and glycerol. So glycerate 3-phosphate, that is the first product of photosynthesis, may be converted to acetyl coenzyme A, which in turn is used to synthesize a variety of fatty acid. So triose phosphate is easily converted into glycerol. So if the glycerate 3-phosphate is converted into fat acids, and then glycerate phosphate is converted into glycerol. So glycerol and fatty acids, they can combine to form lipids, as we discussed in biochemistry, as well as being important storage substance. So triose phosphate, as well as being important storage substance, that is, we are talking of lipids, especially in seeds. Lipids are the major constituents of the cell membrane, and their wax derivatives makes up the waterproof cuticle in plants. Fatty acids produces some flowers scent, that is the smell of different flowers. They smell a good smell so as to attract insects and birds for pollination, which is used to attract insects for pollination. Another uses is the synthesis of proteins. Conversion of glycerate 3-phosphate of Calvin cycle into acetyl coenzyme A is the starting point for amino acid synthesis. So the acetyl coenzyme A enters the Krebs cycle, and from its intermediates, a wide variety of amino acids can be made, and transformation reaction. The amino acids are polymerized into proteins. And another thing is the regeneration of carbon dioxide acceptor molecule. As we studied in the last stage of the Calvin cycle, that some of the triose phosphate has been used to regenerate the ribose by phosphate consumed in the first reaction of the Calvin cycle. The process involves a complex of reaction and requires ATP produced from the right reaction. Requires ATP produced from the right reaction. Here we have some of the study questions. And most of the teachers, they prefer to ask these questions in this part which we have been covered in photosynthesis. For example, they can tell you to write or explain 9 stages of the light or dark reaction. That is question number 1 and question number 2. All of these 2 questions, question number 1 and question number 2, they have answers from Understanding Biology book. So I have lightened the answers here below, but you may go in the Understanding Biology book and then you may understand well, because here I can just write the 9 stages, but there in the book you will see also the diagram which shows those stages one after another until the last stage. And then the third, explain mechanism of the dark reaction, we discussed already. Explain mechanism of light reaction for thickness, and explain how is it important. So mechanism of light reaction, you will be required to explain both cyclic and non-cyclic photophosphorylation, and not only cyclic and non-cyclic, but the whole mechanism as per the last lecture. Explain how Krebs cycle and Calvin cycle they relate, or the elements of each other. When we go to discuss about the Krebs cycle and the respiration, you may understand well, and you may get answers of this question. If the end product of this carbohydrate, explain where the protein and fat in the plant comes from. That is the fate of PGL, and the mechanism is written above. explain the mechanism and give similarities and differences. Similarities and differences between the non-cyclic photophosphorylation and cyclic. I think everybody knows about these questions, but I may jump to the answers of the question number 1 and the question number 2. These questions, these answers they are in the biological science book, but also this is just the mechanism of the reaction. So they have just written in stages. They have just written in stages, but that is the same what we discussed in our mechanism. And if you don't understand anything in these notes here, you may ask me later. But we shall go to discuss later about the part of photorespiration. So in this lecture, let I end here, and let's meet in the next period. Just subscribe the channel for more updates in nutrition. Thank you.